All right. Well, there is one particular molecule that is very widely used as an energy source in cells. And anybody know what that molecule is? It's sometimes called, quote, the energy currency of a cell, because the vast majority of reactions that require energy are use this molecule as an energy source. What is that molecule, folks? ATP, yes. Now, why do we use ATP as an energy source for most of our chemical reactions? Well, let's take a look at ATP. ATP is a nucleotide. You start out with a 5-carbon sugar called ribose, and this is just a kind of standard shorthand representation for it. Attached to this carbon over here is going to be the base called adenine. Okay? And then attached over to here is going to be a phosphatidyne. And then it's connected to a second phosphatidyne. And then it's connected to a third phosphatidyne. In fact, ATP stands for adenosine. Adenosine is the adenine and the ribose. And because it has 3 phosphates, I'm triphosphate. Okay, that's ATP there. Now, when we look at these phosphates are covalently bonded to each other. So covalent bonds are fairly strong. But notice this. Each one of these phosphates has negative charges on it. Those negative charges are going to repel each other because phosphates are actually very compact little ions there. So the charges are quite close to each other. That means they try to repel each other pretty strongly. But the repulsive force is not anywhere near enough to overcome the covalent bonds holding them together. They're like in the days when Alabama used to use chain gangs, which was actually not all that far back when they had fob gangs and stuff. You get 2 people from different gangs that absolutely hate each other and things like that, and they're trying to get away, but they can't because they're chained together. That's kind of what we see here. Okay, now, what we could do is a hydrolysis reaction right over here. Now, this bond happens to be weak enough that it will happen slowly, but it can happen spontaneously at room temperature. It's a somewhat slow thing, but if you have purified ATP, it's best kept in a freezer. The colder, the better, because some of it will spontaneously hydrate. But normally you need catalysts to really speed this up. So if we do a hydrolysis reaction, what we're going to do is we're going to force a water molecule in here and break the covalent bond between the second and third phosphate. So, add H2O, do the hydrolysis reaction. Now we end up with, here's our ribose again. Here's our adenine, I'll just represent it as A. And now we have okay, we have 2 phosphates and we release that free phosphate. Whoops, minus. Okay, now this molecule is called adenosine because it has 2 phosphates, diphosphate. Or ADP. All right. Now, hydrolysis reactions are exergonic. It turns out that because, imagine these guys are trying to pull themselves apart from each other because of the negative charge. They can't break free because of the covalent bond. But now you hydrolyze that covalent bond, and what's going to happen? This guy's going to cut out like a bat out of you-know-where. And in the process, that releases a lot of energy. So ATP hydrolysis ATP hydrolysis is unusually exergonic for a hydrolysis reaction. compared to other hydrolysis reactions, it releases a great deal of energy. Most hydrolysis reactions release a tiny fraction of that. And if you use the molecule GTP with guanine instead of adenine, that releases a comparable amount of energy too as well. Okay, but most of the time we're going to be using ATP as an energy source. The reason we use ATP as an energy source is ATP hydrolysis is generally exergonic enough to provide enough power to do what we really want to do. And that brings us to our next chemical topic, which is what we call coupled reactions. And we're going to use ATP hydrolysis as an example of that. There are other ways of doing other types of what we call coupled reactions, but ATP hydrolysis is the most widely used. The basic idea behind a coupled reaction is this. We are going to use an exergonic reaction in order to power an endergonic reaction. Now, we're going to capture the energy from that exergonic reaction somehow, and that energy that's released is going to be used to provide the energy to do the endergonic reaction that we really want to do. Okay, so we will say a coupled reaction is the use of an exergonic reaction So we use an exergonic reaction to provide the energy to perform an endergonic reaction, which is what we set out to do in the first place. Many coupled reactions, in fact, probably the vast majority of them, use ATP hydrolysis as an energy source. But there are other types of coupled reactions. We talk about ion transport. Sometimes it will let ions into the cell whose transport releases energy. And we'll use that energy to pump ions or other molecules out of the cell in an endergonic reaction. We'll see that later on when we talk about transport across membranes. So sometimes we can use ion flows to provide the energy to pump something else out of a cell, or into a cell for that matter. Okay, we'll see that. That's a type of coupled reaction. But let's focus on the ones using ATP. Okay, let's say what we want to do is take A and B, whatever they are, and join them together to make a polymer. And I'm not doing all the dirty work here. Okay, well, actually what we should do. I'm not showing the details on that, but it's an endergonic condensation reaction. That's what we really want to do. Now, what we can use as an energy source is the exergonic reaction of ATP hydrolysis. Okay. Now, this is an exergonic reaction. Okay, now if we were to kind of merge these two reactions together, we'd have something like this. We take A, we take B, we take water, we take ATP, and now everything reacts together. We get A joined to B, we get a water release from the condensation reaction, we get ADP, and then we get our phosphate ion. Now, for coupled reactions to work, the energy release of our exergonic reaction has to exceed the energy requirements of our endergonic reaction. Now, we're doing here a condensation reaction. What we're using as an energy source is a hydrolysis reaction, specifically ATP hydrolysis. Remember we said ATP hydrolysis releases an unusually large amount of energy for a hydrolysis reaction. What that means is the vast majority of the time, the energy released from ATP hydrolysis is more than sufficient to meet the energy needs of our condensation reaction, or whatever else we're trying to do in the cell. So overall, the summary reaction is slightly exergonic. So you start off, catalyze in most cases, and it goes to equilibrium. Okay. So, ATP hydrolysis generally releases more than enough energy to do almost everything we want to do in a cell through these coupled reactions. Now, once in a while, you run into a case where even ATP hydrolysis is not quite enough. So there's a backup thing here. We could take ATP, and there are a few reactions that are like this, where hydrolysis of ATP still does not provide enough energy. So we got to take this to the next level. What we're going to do in this case is another condensation, or another hydrolysis reaction. But in this case, instead of tearing the third phosphate off, we're going to rip off the second and the third phosphate. So we end up with what's called adenosine monophosphate with a single phosphate ion on it. And then two phosphates covalently bonded together. Okay, that's what we call pyrophosphate. You sometimes see it as a food additive, but pyrophosphate is simply two phosphates covalently joined together. Now, when we do this type of hydrolysis, we're hydrolyzing not just the first, not just the third, but the second and the third phosphates off. That gives you about 50% more energy than just taking the third phosphate off. So there are some chemical reactions, including the reactions that take nucleotides and join them together to make nucleic acids, where simply tearing one phosphate off of ATP isn't enough energy. So in that case, we have to tear two phosphates off of ATP, and that does the trick. Those kinds of reactions are relatively rare, but they are found in cells and biochemistry. Most of the time, hydrolysis of that third phosphate off of ATP is more than sufficient in terms of supplying energy. So that is a kind of coupled reaction. These are coupled reactions. Now, for coupled reactions, you have to do a couple things. There have to be a few points about this. Let's suppose, and I don't have any props here, but let's suppose I wanted to lift this piece of chalk up in the air. Right? Okay, that takes energy. You're fighting gravity. Okay? So that is an endergonic reaction. Now, for an exergonic reaction, let's suppose, now I'm not going to actually put the iPad in this, I get myself in trouble, but let's suppose I have the iPad in, which weighs it's kind of heavy thing. And I were to drop it. That's an exergonic reaction. It releases energy. And a coupled reaction is like, and the energy release from dropping this should be more than sufficient to lift this piece of chalk up in the air. Okay, well, let's do this. Nothing happened. What's going on here? What's happening? Okay, I'm missing something. In order for a coupled reaction to take place, the reactions have to occur pretty much simultaneously. If I hydrolyze ATP, that energy is going to dissipate and not do a thing unless the reaction I want to do is going to happen at about the same time. And talking about milliseconds in most cases. So that's one thing about coupled reactions. The reactions have to happen pretty close to simultaneously. Okay. Second thing, the reactions have to happen physically close together in the same space. Even if reactions occur simultaneously, if my exergonic reaction occurs here and my endergonic reaction is way over there, the energy is not going to get transferred over to there. So they have to occur in the same physical space, very, very close together. Now the third thing about coupled reactions is illustrated by this. Doing this exergonic reaction, nothing is happening. Now suppose I put something like a little seesaw or lever arm here, where I can transmit the energy from this to this. In other words, I physically join the two reactions together. Now you may remember from your childhood things like seesaws and stuff. Okay, somebody gets on, or sometimes you saw at circus performances, somebody's on one side of a seesaw, somebody stands up on, a ladder or something, and jumps off and hits the thing, and the clown goes flying up into the air. Now if somebody took the seesaw away, all that would happen is the guy who jumped off the ladder would break his leg or something like that, whatever the case would be, right? Nothing else would happen. So you have to have something that physically joins the two reactions together. So the energy is transferred from the exergonic reaction to the endergonic reaction. You need some kind of connector. Now, the seesaw type of thing is a physical analogy to that. But what happens here is what joins these reactions together is the active site of an enzyme. That's like the seesaw. Now, by the way, although I don't have the props here to do that, if you look at YouTube, I have a little mechanical demonstration of it, and where I get to destroy the podium in the process. That was not intentional, but I have a mechanical analogy of a coupled reaction. And the end result, some of the excess energy not only went to do what I wanted to do, it also went to destroy the podium. That's up there on YouTube. It says something like Dr. Sauterer destroys the podium classroom demonstration. Okay, normally it does not happen, but that shows you, illustrates a point there, so I recommend looking. It is pretty funny to watch me ducking his pieces or flying all over the place. But occur on the active side of the enzyme. The active side of the enzyme joins these two reactions together so the energy can be transferred from the exergonic reaction to the endergonic reaction, and both reactions can occur simultaneously. So here we have an enzyme, and let's suppose our old thing here, we want to join A and B together. So part of the active site binds ATP. And part of the active site binds our two substrates A and B. Part of the active site hydrolyzes the phosphate off of ATP, generating ADP. And then the energy released from that is now used by the enzyme to catalyze the covalent bond between them. In other words, do the condensation reaction. As a matter of fact, what actually happens in these cases is you transfer the phosphate ion from the ATP to one of the substrates, and then you carry it on from there. So in this case, an enzyme that is going to do something like joining together two sugars to make a polysaccharide, that enzyme is going to bind ATP as an energy source. It will hydrolyze the ATP, and in a fraction of a second, that phosphate gets transferred to one of the substrates, the condensation reaction occurs, and now you've got those two substrates joined together. The enzyme puts them all in the same place, makes sure the energy gets transferred from the exergonic to the endergonic reaction, and makes sure that they occur in the proper time. In other words, virtually simultaneously. Okay, so that's coupled reactions, very important in all kinds of biochemical processes. And like I say, although we use ATP hydrolysis here as an example, there are other kinds of coupled reactions as well. We'll find the process of oxidase metabolism or oxidase phosphorylation is another kind of coupled reaction where we're using the energy of oxidation to move ions from one cell compartment to another. We'll see examples where you move ions in one direction, releasing energy, and then we use that energy to move something else in another direction in an endergonic process. So the ATP hydrolysis is not the only type of coupled reaction around, but it's a widely used one. And that's why we use ATP for the vast majority of reactions that occur in cells. Okay.